An overview on post-translational modifications. Just as post-transcriptional modifications gave a variety of structural and functional diversity in the transcript that are synthesized or processed. Similarly, in post-translational modifications, due to the additions of various chemical moieties, the genome can give rise to a much complex and diverse proteome. So, the variety of proteins that are obtained from a single gene is amazing to note because through post-transcriptional and post-translational modifications, a single gene is able to probably give rise to several different proteins. Now, let us therefore look at the outcomes of this session. So, the post-translational modifications occur mainly in the eukaryotes, with prokaryotic seeing a very limited number of post-transcriptional translational modifications. And this is basically in order to diversify the protein structures and functions and coordinate their signaling networks. These modifications are a range of chemical alterations, and mostly they are enzymatic. And what has been observed is that these modifications can aid in catalysis, in regulation, and in enhancing protein folding and much more. So, therefore, due to these modifications, the proteome is much more complex, just as I just mentioned, and then than the genome of a cell. Defects in these mechanisms lead to various disorders and diseases. So, therefore, you can note how important these modifications are. These modifications may be reversible or may be irreversible depending on the kind of modification that has taken place. So we will look at what are the different post-translational modifications. Do note that not all modifications have been covered, but only the ones that are more prevalent. So post-translational modifications, phosphorylation is one of the most common uh, post-translational modifications. Glycosylation is another modification that is very prevalent. Then you have acetylation, you have smolylation, lipidation, S-nitrosylation, ubiquitination, and proteolysis. These are the ones that are more common. But there are many others. So, for example, hydroxylation is also a post-translational modification. So, similarly, there are many different types of post-translational modifications. So, we are going to get an overview of some of these post-translational modifications just to get an idea of how these modifications impact the proteins. So let us first look at phosphorylation. So what is phosphorylation? Say for example, we have a polypeptide, and when this reversibly, so it can either get modified and then get back to an unmodified form again. So it reversibly gets converted into a phosphorylated uh, protein. So you can see the phosphate moiety being attached. So the attachment of the phosphate moiety is mainly because of kinases. These are enzymes that either use ATP or ATP to add a phosphate moiety to a substrate. And there are phosphatases that enable the backward reaction. And these phosphatases are enzymes that remove phosphate from a substrate. So that is how this modification is a reversible modification. It is possible that it becomes a reversible modification. So what mainly happens when a phosphate moiety or phosphorylation occurs? The first thing that is observed is that it brings about a conformational change. Plus, because the phosphate has an overall negative charge, okay, and there are electrons present which can interact with surrounding amino acids or the microenvironment within the polypeptide. There are functional attributions because of this phosphate. So you may have a inactive protein getting active because of phosphorylation, Or you may have an active protein getting converted into inactive protein because of the phosphorylation. Well, it can also be that when phosphorylated it becomes active, and when the phosphate is removed it can become inactive. So that way also it can be reversible. Plus it also can be so that an active protein which is in the phosphorylated form, when the phosphate is removed it can become inactive. So this is something that is extremely important, and it plays a very regulatory role, especially in metabolic pathways and many molecular mechanisms. So the main kinases that have been observed are serine kinases, threonine kinases, and tyrosine kinases, which means what? That the phosphate moiety is added to the OH of serine, OH of the threonine, and OH of the tyrosine. So that is how the phosphorylation takes place. So these residues mainly in the polypeptide are responsible for getting phosphorylated. Now let us look at an example and see how the impact of phosphorylation is. So when you have glucagon coming and binding to the hormone receptor, okay, uh, through a cascade of signals, what happens is that the glycogen synthase, okay, which is in A form, that is, it is active. So without the ATP, it is in the active form. But suppose due to a protein kinase, so you can see this is a kinase, which means it is going to add a phosphate group to the glycogen synthase, as glycogen synthase is its substrate. Okay, the glycogen synthase is phosphorylated. So now you have a phosphorylated glycogen synthase. And this phosphorylated glycogen synthase is now in an inactive form. So therefore, a phosphorylated glycogen synthase is not going to synthesize glycogen. Why? Because it has been phosphorylated. Now, at the same this thing, if you have a glycogen synthase which is phosphorylated getting dephosphorylated because of a protein phosphatase, then it comes back to its active form and now it will start synthesizing glycogen. Now, so this is so relevant. A glucagon is responsible for uh, you know, due to hypoglycemic levels in the body, it is responsible for releasing glucose or releasing sugar into blood. So, in cells, especially target cells, you have the sugar stored in the form of glycogen, say, for example, in the liver. So, in order to get glucose, if there is a large amount of sugar present, then these glycogen synthases are responsible for glycogen synthase is responsible for synthesis of glycogen from glucose. Okay, so an active glycogen synthase is going to synthesize glycogen. But what does the glucagon do? The glucagon is inducing the activity of protein kinase, which is able to phosphorylate the glycogen synthase. Why is this important? Because if glycogen synthase is inactive, then it is not going to synthesize glycogen. So sugar is not going to be used to synthesize glycogen. Rather, the sugar can be given back to the blood to increase the sugar level in the blood. Or what can happen is that because this is inactive, you can have another enzyme called glycogen phosphorylase, which in turn, when it gets phosphorylated, becomes active and it degrades glycogen into glucose 1 phosphate. And that glucose 1 phosphate is converted to glucose and then released into the blood. So you can see how just a simple phosphorylation is activating or inactivating an enzyme, and that has an interesting physiological response. So therefore, you have many enzymes in the metabolic pathways or even in molecular mechanisms that when phosphorylated are either active or inactive, and when the phosphate is removed it becomes active or inactive. Another example is that of cyclin-dependent kinases. They themselves are phosphorylating many substrates, thereby regulating cell cycle. So this way there are many proteins that on phosphorylation not just change conformation but also change their own functionalities. Then we go to glycosylation. Glycosylation is by basically addition of a single sugar molecule or oligosaccharide, or you would have a specific moiety that is being glycan moiety that is being added to proteins. So such additions of sugar moieties to a substrate is what is called as glycosylation. And we all know that glycosylation majorly happens in the endoplasmic reticulum of the cell and the Golgi body of the cells. So how does it help? It helps one in proper protein folding. And if protein folds properly, its native conformation will be obtained, which is responsible for enhanced or optimal biological activity. So that is what is observed. And of course, the conformations can change depending on the glycosylation. The glycosylation enables a molecule to become more stable, and it also helps in distribution of these molecules. So when you have a particular sugar added to a particular protein, it may end up in mitochondria. But when you have another sugar added to that same protein, the protein can go to another organelle. So therefore, protein sorting is helped by glycosylation. And therefore, many of these glycosylations that happen in the ER and in the Golgi through endomembrane network pathway, a lot of proteins are sorted to respective organelles. It may be either lodged in the plasma membrane, or it may go to the lysosome, or it may go to a proper, or it may just get secreted out. So depending on where the protein has to go, their glycosylations can be different. So what are the types of glycosylation that is possible? One is N-glycosylation. This is observed particularly in the endoplasmic reticulum, but not just in the endoplasmic reticulum. Many other proteins and other organelles are also N-glycosylated. N over here stands for addition of a glycosyl moiety to the NH2 of an amino acid residue. So for example, you have arginine, and arginine has as its side chain an NH2 end, and if a glycosyl moiety is added to the N, then it is what is called as N-glycosylation. So as you can see, on a NH2 moiety, you have the glycosyl group being added. O-glycosylation. So when the glycosyl moiety is added to an OH group, either on a serine or a tyrosine, then you would have what is called as O-glycosylation. Glycation. What is glycation? When you have the sugar moiety that has, or that is already attached to a attached to a phosphatidyl moiety. So it is basically a phospholipid with a sugar moiety. So when this is added to the protein, then that is what is called as a glycation. So now what has happened is that a lipid moiety is also associated with the protein along with the sugar. So the protein through the sugar moiety is attached to a lipid moiety. C-glycosylation. What is C-glycosylation? When the glycosyl group is added to a carbon, say for example of an aromatic amino acid residue. So we know that all aromatic amino acids have a ring structure which has primarily carbons as the skeleton. And if the glycosyl group is added to those carbons, then it is what is called as C-glycosylation. So for example, you have the indole group of the tryptophan. So if the glycosyl moiety is added to the indole group, then that is what is called as C-glycosylation. You have what is called as phosphoglycosylation. So here again what you can see is that the sugar that is attached to the phosphate moiety of a phosphatidylglycerol, okay, or it is attached to a phospholipid, then that when it attaches to the OH group, that is what is called as the phosphoglycosylation. So you have these many different types of glycosylation possible because of different enzymes present in a cell. Now, let us just take the example of an antibody. If you can observe over here, you have a oligomannose type end-linked glycan attached, which is present between the two heavy chains, the constant regions of the two heavy chains. If, say, for example, the enzyme that is glycating at this position is defective and this glycan molecule is not attached, then what has been observed is that this entire constant region of the antibody collapses. And if that collapses, then the functionality of the antibody is going to get affected. So, as the means of glycation, you can see that the molecule is getting stabilized. And because it is getting stabilized, it has a activity that is optimal. So therefore, this was just an example to show how glycosylation helps. Then we come to acetylation. Acetylation is addition of an acetyl group. So it can be end-linked acetylation, it can be reversible or an irreversible mechanism as well. So if it is a reversible mechanism, then there are deacetylases that are able to remove the acetyl group from the substrate and thereby getting it back to a condition where it does not have an acetyl group. Acetylation in proteins can help cross-talk with other post-translational modifications. What does that mean? It means that if a protein is acetylated, then this acetyl group can interact with the phosphate moiety on another protein, or it can interact with a glycosyl moiety in another protein. So therefore, this acetyl group is able to interact with many other moieties that are used to modify proteins. Acetylation of histone tails is a very good example of post-translation modification by acetylation. And what is very important because of acetylation of histone tails of the nucleosome is that they play a very important role in gene expression. So let's consider, for example, you have the DNA wound around the nucleosomes, that is the histone octamers, and this winding around of the DNA on the nucleosomes, and then the nucleosomes with the DNA further folding or further getting into more condensed form. So here what you observe is a solenoid form. In this solenoid form, what is very clear is that all the DNA is wound around the nucleosome and they are not accessible for expression. So you cannot have an RNA pol going and binding to a promoter which is ensconced within these nucleosomes to enable synthesis of a transcript. Now, suppose for example, you have histone acetylase that adds the acetyl to the histone tails of these nucleosomes. Now, acetyl. So, please note, before I come to that, please note that for DNA to wind around histones, it is basically its negative charge and the histones positive charge that enable them to interact and make it a stable interaction. Now, if an acetyl group is added, the acetyl group ends with COCH3. So the CH3 does not have a charge on it, and therefore what happens is that on the histone, the positive charge is neutralized. So because of less positive charge being present due to acetylation of the histone tails, the interaction of the DNA with the histones is going to get minimized. And so the DNA is going to be loosely packed around the nucleosomes and therefore they will be more accessible for expression. So an RNA pol can definitely come and bind over here, you see, to start synthesizing the RNA from the DNA. So therefore, when the histones are acetylated, the chromatin is much more relaxed, and all of us know that if chromatin is much more relaxed, then it allows for a greater gene expression. So therefore, that is how it is important. Now, for example, if you remove these acetyl groups again, the relaxed chromatin will become a closed chromatin. And that is by what is called as the histone deacetylases. So if you have inhibitors for histone deacetylases, then the chromatin will always remain as a relaxed chromatin. So again, what you can observe over here, that simple acetylation and deacetylation can influence gene expression and thereby the function of the cell itself. So deacetylation can cause what is called as gene silencing. Now we look at another modification, which is methylation. So N or O linked methylation is what happens, and N-linked methylations are considered to be irreversible, while O-linked methylations can be reversed. So it increases generally the hydrophobicity of a protein as methyl ends are non-polar. So you can again see that if a CH3 group is added, CH3 has no net charge on it and therefore it becomes a non-polar group. So thereby more methylations, more the hydrophobicity of a protein. So you can see that the nature of protein is getting changed because of the hydrophobicity and therefore the conformation of the protein will get changed. So methyl transferases transfer methyl group specially from S-adenosyl methionine. So this becomes the source of the methyl group. As you know that methionine has a methyl group present on it. So to target the amino acids, from these you get the methyl group. The amino acid residues may be added more than one methyl group. So it's not necessary that only one methyl group is added, but you can have more than one methyl groups also being added. Methylation is known again to bring about epigenetic changes. Uh, what is epigenetic change? Now, when a nucleotide is getting methylated, okay, then the methylated nucleotide may not be recognizable, say, for example, by an enzyme. So it is basically an altered nucleotide. So the methylation has changed its look. And so even without a change in the sequence of nucleotides, so the nucleotide itself is not changing. So it's not becoming from adenine, it is not becoming a thymine, but it is adenine itself. However, the adenine becomes say methyl adenine. Now, methyl adenine, the look is different from adenine. So, hence, such kind of changes are what are called as epigenetic changes. And again, epigenetic changes are responsible for influencing gene expression. These epigenetic changes may be inheritable, may not be inheritable. So it may only be for a specific generation and doesn't go to the next generation, but there are some epigenetic changes that may also go to the next generation. So as you can again see over here, you have the histones getting acetylated at various lysine moieties. Okay, so this is again example of the histone tails getting methylated by the methyl group. But you can have methylation on various other proteins also. This is just an example. Lipidation is the next post-translation modification, as the name suggests, addition of a lipid moiety to a protein. So for example, because of addition of lipid moiety, you get lipoproteins, or you may get a protein with a lipid moiety and that lipid is helping it to be embedded in the plasma membrane or embedded in a membrane structure. So many uh, extrinsic proteins, okay, are bound to the membranes through such lipid moieties. So it is a way of targeting proteins to membranes. It increases hydrophobicity and thereby relative affinity to bind to membranes. So lipidation can be through GPI anchors, can be through myristylation, can be through palmitylation, and can be through prenylation. So this is showing the different ways by which lipidation can take place. Now, when you have in the plasma membrane, as you can see, a phospholipid, so this is basically a phosphatidylinositol. When the phosphatidylinositol is glycated, so this is what is called as GPI anchored protein. So this becomes the glycosyl phosphatidylinositol anchored protein. So this protein is basically an extrinsic protein, but it is linked to the membrane through this GPI molecule. So this becomes the glycosyl part, this becomes the phosphatidylinositol part, this becomes the anchored protein. Okay, so that is how you can see that how the protein is targeted to a membrane. What is now myristylation? So for example, suppose you have AMP kinase. This is just an example of a protein that is being synthesized by the ribosome. And as it is being synthesized by the ribosome, you have the myristoyl transferase transferring, so N myristoyl transferase transferring a, uh lipid moiety to the protein. So therefore, when it gets lipid moiety, then it is transferred to the lysosome, and when it is transferred and attached to the membrane of the lysosome, it has been observed to inhibit toll-like receptors which are responsible for a certain immunological response. So this is going to affect the immune response. Just simple myristoylation is going to affect the way the immune response is going to be. Now, myristoyl is just a 14 carbon lipid moiety, and therefore it is not necessary that this AMPK or any protein which is myristoylated becomes a permanent part of the membrane. It is there only temporarily. Then you can have palmitylation. So when a palmityl group is added to a protein, and generally it is S-palmitylation, so a DHHC enzyme is responsible for adding the palmityl group to the cysteine of a substrate, while you can have this reversible by a thioesterase that is able to remove again the palmityl group from the cysteine moiety. So this is how you can have the palmityl group added and the palmityl group removed. Similarly, prenyl groups. So you can have a farcinyl or you can have a geranyl molecule that is added to a substrate. This is also S prenylation. So it is added to the cysteine thiol group, and that is how a protein will become farcinylated, or it will become, sorry, farnesylated, or it is going to become geranylated. So this is how isoprene molecules are also added, and you all know that isoprene molecules also belong to the non-polar groups of molecules. Now we go to ubiquitination. Ubiquitination, as the word suggests, is addition of a molecule of ubiquitin to protein. And why is it named as ubiquitin? Because it is present in all cells everywhere. Now, ubiquitin is a very small protein of 76 amino acids, and through a glycine at its C terminal, you have the ubiquitin being added to an amine group of lysine in the target protein. So it is already always added to the lysine. And this is thanks to what? This is thanks to three enzymes that are able to carry out a three-step mechanism to ensure that a ubiquitin is added. But you can observe that ubiquitination is something that not one ubiquitin is added, but several ubiquitins can be added. So as you can see here, this is the protein. Okay, by the use of E1, E2, E3, these are the three enzymes, you can see a ubiquitin chain has been added. And when a protein is generally ubiquitinated, the ubiquitinated protein is targeted to a proteasome, and within this proteasome, the protein is degraded. Ubiquitin is released again, so you can see the protein is completely degraded to amino acids, okay, or to smaller peptides. So therefore, ubiquitination has an important role in actually degradation of proteins per se, and also targeting it, say, for DNA repair, etc. So therefore, ubiquitination is also an important post-translational modification. Now we come to another post-translational modification, and that is proteolysis. That is removal of a part of the protein. So it is basically enzymes that can break peptide bonds that are that carry out proteolysis. So these enzymes are proteases. They can be serine proteases, etc., etc. So many proteases in the cells with specificity, of course, it goes and cleaves only specific proteins, and the proteolysis could lead to various functionalities. So you can understand that there are thousands of proteases that are present in within one cell. Proteolysis is thermodynamically favorable and it is generally irreversible. As they cleave peptide bonds, these enzymes are under tight regulation. So they cannot, since it is an irreversible process, they cannot be not regulated, they are regulated. Their classification is based on their site of action. Now, say for example, you take insulin. So when insulin is synthesized, it is synthesized in the pre-proinsulin form, and in the pre-proinsulin form, you have the signal peptide, you have the A chain, you have the C peptide, and then you have the B chain, C peptide, and the A chain. So through proteolysis, the signal sequence is first removed. So you have a specific signal peptidase which will remove the signal sequence to leave the proinsulin. Now once the proinsulin is formed, you can see that intra disulfide linkages and are formed to stabilize the structure, and the A chain is attached to the B chain. Then you have another proteolysis which removes the C peptide. So what remains is then the A chain that is attached to the B chain, and this is the final functional insulin. So you can see how proteolysis leads to an inactive pre-proinsulin to an active insulin. Conclusions: post-translation modifications of protein can lead to diverse structures and functions. There are many types of modifications with more than one possible for each protein. What does it mean? If a protein is phosphorylated, it may also be acetylated. If a protein is methylated, it may also be acetylated. So more than one type of modification is possible in one protein. And the perfect example for this is histones. Phosphorylation is observed across many species, and they are known to function as a mode of regulation in many pathways, especially signal pathways. Glycosylation is one of the most common post-translation modifications that especially are responsible for rendering a very defined enhancement in the biological activities of many molecules. Lipidation, ubiquitination, hydroxylation, proteolysis, etc. are all modifications that create a proteome that is beneficial to the cell. Thus, post-translational modifications, if not done as per the requirement, then there are several important physiological, molecular, metabolic pathways, etc., that can be disrupted, leading to diseases and disorders. Thank you.